All right. Welcome back, everyone, to the third session in our first day of the second part of the Science of Cannabinoid Cultivation Academy. So we've got Leighton-Morrison here, the man in person, to give you some knowledge on AMOs. So I just want to kind of remind everyone, in case you missed the earlier sessions, some standard stuff. We've got a chat window over on the right-hand side. You can chat in there. You can also post questions, and we'll be collecting the questions and saving those for later on in the session. It's going to be a 2-hour session, so about an hour and a half presentation, half an hour of Q&A. So start those questions coming, and we'll get to them towards the end. And just want to let everyone know our sponsor for the whole event here, our title sponsor, is AgroCoco. They have some really high quality cocoa coir products. And our sponsor for this session is BlueMat irrigation systems. So I think without further ado, I'll just turn it over to Mr. Leighton-Morrison here.
All right. So let's take a deep dive. What are AMOs or otherwise AMs? We all know, well, hopefully you know that an EM is an effective microorganism. An AMO or an AM is an aquatic microorganism. A lot of you out there, I'm sure, like, well, what do I need AMOs for? AMs. They actually play a very vital role in our soil systems, and we're gonna go down that rabbit hole. So, why do they matter? You've heard Chris say it. You're going to hear this repeatedly. It's all about diversity. And there were some questions last time about getting these minor cannabinoids, cannabinols, terpenoids, terpenes to be more prevalent. And in the work that I've been doing over the years, it's very apparent that diverse soil is the result or is the driver for getting those expressions, those minor expressions, to be much more pungent and available. So I mentioned IMO collections. I actually talked to Chris about this yesterday. You know, can IMO collections be done in an aquatic microorganism to collect microorganisms? And the answer is no, because you can't soak your rice in water. It will go anaerobic and mushy. I will make some suggestions further in this presentation about how you can do that. So the next question is food versus colonization. Can these aquatic organisms colonize the soil system? And the answer is yes. And I will, again, get into that in detail as we move forward through this presentation. And again, to remind you, soil is what? In my last presentation, cyclical soil, I talked about soil only being 50% physical, and that leaves a lot of 50% left, which would be air and water. So we'll get into that a little bit further, and then do these nutrient cycle. Or excuse me, I skipped one. What lives in water? And this is an interesting question. I wish James had more time. He and I have talked at length about how these endophytes actually go through phases similarly like insects. So if a endophytic bacteria is in a leaf, it falls from the tree and lands on the floor of the forest, it goes through a saprophytic phase. So it starts to grow out and consume and decay the matter or the leaf itself. What happens if that leaf falls into water? Well, it actually comes out and becomes an aquatic fungi. So there's a lot of this stuff we don't know. And this is going to get really confusing later in this presentation because so many of these organisms have been switched around. As science catches up with our understanding, we're constantly shifting what we call kingdoms around. And kingdoms are the overall guide of where organisms go, and then their species and taxonomy and all this other stuff. So we're not going to go too crazy down that, but just understand that as this knowledge comes out, we do the best we can to update our information to keep you guys relevant. But again, with all the science that's going on, there's going to be more and more changes to this. There's no doubt in my mind. So these organisms can live in soil and they can live in water. And again, what is soil? Soil can be up to 50% water. So think about the possibility of cross-pollination between these organisms. Do they nutrient cycle? Absolutely. It's pretty apparent that the nutrient cycling system in the aquatic food web is as or not more intense than in the soil food web. And that's because most aquatic animals, for instance, fish, go through a 3-step phase from a larval stage to a fry to a fingerling. And then from there on, as they grow bigger, they're considered fish. So during those different life cycles, they're eating different things, and they start by having that egg sac that provides them the nutrients. But as soon as that egg sac starts to dry up, the first things they're after is rotifers, as a main staple food, and that is a aquatic microorganism. And I've seen them in soil as well, so I know there's quite a bit of crossover. So again, I think that we will see as we get more and more down that rabbit hole, that the aquatic food web can be, if not more so, complicated than the soil food web. All right, I want you to look at that slide for a minute, and then I want you to just kind of close your eyes and try to visualize that picture in your head. As the great human expansion took place, we'll call it starting 2,000 years ago, where we really started to multiply exponentially, therefore consuming more resources. We affected the migration of a lot of things. So even when I was a boy living in New England, we would have these incredible V's of Canadian geese traveling up and down the coast in the spring and the fall. And so as a migratory bird, of course, they're stopping every so often to drink and eat. And then when they lift off again, what do you think's dropping off of them? Tiny droplets of water containing potentially thousands of aquatic microorganisms. Again, think of the four-leggeds, buffalo, water buffalo, deer, moose, bear, all tromping through rivers, lakes, streams, and dragging that soaking wet hair up onto the shore and having it slowly drop off. And then you add a predator to that, so a lion or a bear perhaps, chasing you. You're gonna get those organisms spread a lot further inland. But again, also think of back up to the birds. Songbirds migrate thousands of miles, and they're constantly stopping to drink and eat. We know for a fact that bird eggs will survive in the belly of a bird, and therefore if they poop over a lake, river, stream, that fish egg will become viable again because it's back in its aquatic environment. So those winged and four-leggeds were a huge impact on transporting microorganisms across the planet before we stopped that in many ways. And we expanded beyond their ability to have the land space that they needed. Fish, okay, finned animals. They are eaten by a lot of predators. And when they're brought up on shore to eat, not everything is eaten. Inside their gut is an entire aquatic ecosystem in its own. And of course, on the outside, in the gills and scales and their fins are all caked with aquatic microorganisms. So there's another delivery system to getting these organisms up onto the land. Then we have amphibians and reptiles, and these guys go in and out of water all the time. And so again, these guys are more closely associated to the edges or the riparian system. But if you have a riparian system where one of these amphibians or reptiles are captured and or eaten, I should say, and dragged further inland, again, you're having an inoculation, so to say, of the soil around them. So this is important. Water and soil. And again, I said only 50% is physical. The other half is water and air. So if you have too much air, you're aerobic or anoxin. And if you have too much water, say that whole space was filled up with water, you would be anaerobic or, again, anoxin, so low oxygen content. Why is that important? Well, you'll hear me talk about aggregation a lot. Soil aggregates are the key to this whole thing. So imagine a microclimate inside an aggregate where you have, little condensation drops, you have little pockets of water. You'll hear a lot about, people that do soil work will tell you there's anaerobic pockets and there's aerobic pockets. There's anoxin pockets. So you're going to have a combination of all these different things. So if I inoculate soil with an AMO or an AM, what is the likelihood that those organisms will stay in function? And if they're in one of those water droplets or sucked into a macro aggregate, the chances of them surviving is pretty good. And again, I've seen, this is anecdotal. Well, it isn't. I have done testing on projects I've done over the years, and I have seen increased levels of flagellates, amoebas, diatoms, and some of these other organisms that were not present when I started the process. So I would, on these jobs, I would take soil samples, look at what's there, and of course do the inoculations, and then come back a year or so later to see how well they worked and if the fields or the soil systems are holding up. So I know, again, that this is a viable solution to getting some of that diversity back in the soil. So anyway, aggregation are the key to the whole thing, and they are basically have the ability to store these organisms if they have a good aggregation. Aggregation is a way to measure how functional the soil is, because if you have aggregation, that means you have a lot of glues. So the glomalin from the fungi and the biofilm from the bacteria. Let's talk about water for a minute. RO water. I think everybody knows about reverse osmosis. So what reverse osmosis does is basically squeezes a water molecule through a rubber glove. And that's pretty much, a good way to look at it. Inside a single cell bacteria, there is about 3.3 billion water molecules. So you can tell they're pretty small. And again, when you use RO water, you've stripped everything out of it. So the good things as well as the minerals and the bad things. So what is the best source of water? In my mind, it's spring water. So this can also be called an artesian well, where the water just bubbles up through the surface of soil. Where I get the AMOs that I work with come from a place on Cape Cod where literally you just take the end of the shovel and you stick it in the soil and water will start bubbling up. And so that is super minerally rich water. It's very clean because it's had a ton of time to come from Canada, southern Vermont, and northern New York is where it's been determined that this aquifer starts and pretty much goes straight down to Cape Cod, which is a sandbar, and bubbles up as a river in West Barnstable. And I highly suggest you Google Earth it. It's a great example of what a really good spring water looks like. Now let's talk about well water. Whenever you drill a well, you punch a hole in Mother Earth. And so the way water usually gets down into an aquifer is it takes a long time. First, it goes down into the ground, pools into what we call groundwater, and then slowly over time migrates through these tiny little fissures and cracks in the stone or the bedrock, and eventually gets down into loose gravel sand where it enters into the aquifer. Traditionally, that has been a fantastic source of water. But since there's so many people and this great expansion has occurred, everybody punches a well. Problem is that contaminants can slip down around the outer casing of that well and actually, pollute the aquifer. And I've seen this a number of times in highly agricultural areas where the well's out in the middle of the field. They're, fungicides, they're fertilizing, and the cancer rates tend to go through the roof. And I know here in California, the Central Valley has huge issues with the well water. And now they pretty much use all of the water they bring in from the Colorado River. So moving on, we talk about town water. Town water is chloramine. So chloramine is the combination of ammonia and chlorine. And so when those two come together, they form an ionic bond, which is stronger than a covalent bond. And so it's very difficult to break. The only way to break it is to use a humic acid, not a fulvic, but a humic acid. And what that does is it breaks that bond, and now you can bubble off or aerate the chlorine to get it to go away. And the ammonia is now a product that can be consumed by bacteria, as Chris talked about, and is a source of food for the biological life. So what is AMO water? Well, if you can support fish, you got to have pretty good water. So just the aquatic chemistry alone, if any of you have kept fish at home, you know a little bit about this. pH is really important because pH regulates which minerals are available and which ones are tied up. So if you have a fish tank, you're pHing all the time. And if your fish are logy and unhealthy, that's probably due to your water quality. And if their fish are vibrant and their colors are, vivid and they're bombing around the tank, you got really well-balanced water. In my work back at Rodale, I had a 1,000-gallon aquatic system or aquaculture system, and I would get the water to this like tea. It was just like this light, pale yellow water. And whenever I got that to that water quality, the fish were just so vibrant. And so this way of using fish to fertilize soil and plants, it's called aquaponics. If you're getting that level of healthy fish, you're going to have super healthy water. And so the water that the AMO that I'll discuss later in this presentation comes from a source just like that. And as I said, it comes from an aquifer that travels, hundreds and hundreds of miles and comes out in Barnstable, West Barnstable, Massachusetts. So fish waste biology, this pie is, I wish it was clearer. I wish I could have blown it up a little bit. But if you look carefully, it's very complex. And these are all communities. And some of these communities have different relationships with other ones. Hence the colors kind of shows you the scale of all of this. Now, are all of these going to colonize or to inoculate soil? Probably not. Some of these are just going to be foods because they're not going to live, for long periods of time in a soil system, because as it dries out, they probably can't take the pressure. But I wanted you to see this slide because I think it's really important in showing just how crazily diverse a fish waste biology cross-section looks like. Incredibly diverse. Aquatic bacteria. So autotrophic are primary. So basically what they can do is they use sunlight, they mine minerals, but they make their own foods. Heterotrophic, these guys eat the primaries, the autotrophs. So I want you to understand there's a prime example of a cycling system, and it just gets more and more complex as we get further down this rabbit hole. Our cyanobacteria algae, they're a complicated one. They are, I call them the misfits because they fit into so many different categories. They called them bacteria at the beginning. In my mind, they probably should have dropped the bacteria, but then again, other bacteria have this problem of falling back and forth between multiple kingdoms. And that's why I warned you up front that it is going to get a little bit confusing. But just bear with me and try to learn as a bigger picture and not necessarily try to not nitpick, but like focus on one thing or the other. So another great aquatic bacteria is nitrifying bacteria. You heard Chris talk about, I forget which species, but in the aquatic ecosystem, you have a tremendous and diverse community of nitrifying bacteria, as well as phosphate solubilizing bacteria. There's some products on the market, which I know most cannabis growers know all about. They are grown in vitro. So that means they're grown in a lab. And as Chris said, bugs in a jug, yeah, they work. They'll give you a bump, but you're going to have to replace them. They will not colonize. And these organisms that come out of a in vivo system, so in living system, definitely colonize, and they definitely remain in play. And then a lot of people don't know, but there's potassium solubilizing bacteria also in an aquatic ecosystem. So right there is your three majors: nitrogen, phosphorus, and potassium. Easily put into play in a soil system and can mine material for you indefinitely, as long as, again, like Chris said, you don't let it completely dry out, because you're probably going to lose some of this diversity. Good news is you can still get more of them if you need them. Are they terrestrial too? And here again, the answer is yes, because a lot of these waterways, like the ocean, you have high tide, low tide. And also you have heavy rains in spring and fall, and virtually no rain in summer. And of course, you have snow in winter. So they're going through these constant, what do I want to call, wet and dry phases. I'm not saying that they're all going to survive that, but they will definitely cyst up and support themselves in that system until the rain comes again. And lastly, biofilm. Again, you heard me talking about aggregates and their critical value in soil. You will never achieve a healthy soil system if you don't have aggregates, because basically you have the components, sand, silt, and clay, and organic matter, just piled up on top of each other. So therefore, you will have no water infiltration. So how are you going to establish, a healthy infiltration and oxygen exchange or gas exchange? You can't. Without those aggregates, you're going in the wrong direction. And biofilm is critical to form aggregation. So even if these organisms only lasted for a short period of time, they are going to produce a tremendous amount of beneficial biofilm for your aggregates and potentially build the entire aggregate even bigger. All right, aquatic fungi. They're heterotrophic, and so we also, in the water system, we have endophytes, saprophytes, mycorrhizae. A lot of people don't understand that mycorrhizae was perhaps the first fungi to come out of the water, fresh water, and up onto the shore to colonize and make mushrooms, which many people believe. And again, we may prove this wrong, but that the fungi were the first plants to be on the surface, and then as they grew like incredibly fast and then died, they were the beginnings of organic matter, which allowed plants to come bigger and more diverse plants to come next. You also get a tremendous amount of yeast. You've all heard of water molds, zoospores. That's something that again, it's a crossover, so we have to be a little bit careful. And there's also fungi that have flagellates, and that's something else that a lot of people don't realize, is that flagellum are on many different types of things, and not only fungi, but bacteria, and we all know about protozoa, which both the flagellates and the ciliates have them, and a lot of others as well. So protozoa, a lot of people already know about flagellates and amoebas, but there's also cyanobacteria. They have a flagellum and they move. And blue-green algae, people think of as, red tide, toxic to fish, but in a balanced soil system or a balanced ecosystem, they're part of it. They're important. They play an important role as a cycler and as a food source. And a lot of people don't realize that, yes, green and red algae also can have flagellum, and therefore they're modal. They move through the system. And also terrestrial. So, in collecting AMOs and studying them for over 12 years now, actually more since I was kind of a kid, I've seen this constant transition from taking, the fish waste out of my little fish tanks, squeezing it into a bucket of water, adding some more water, and then stirring it up, and then putting it on a plant. You just see incredible plant reaction. So I truly believe, and again, this is a hypothesis, I don't have any hard data, although I have seen some white papers, not a lot, explaining that a lot of these things can be, they can go back and forth between soil systems and aquatic systems. And I've already seen that with flagellates and amoebas, not so much with these other fine algae. But I'm sure it happens. So algae, there are a ton of these, and we are just really beginning to get a grip on how valuable these algae are as far as nutrient cycling. Think of kelp, a macrophyte. So that's a giant plant underwater. They grow 30 feet high, and it's an algae. It's living in a very hostile environment, very little oxygen, not a ton of nutrients. Although the saprophytics that have mathophytes, butchered that one, help to provide nutrients for these kelp. And of course, the cyanobacteria, here we are again, blue-green algae, all of these are, autotrophic protists. So, and they can go down to single cells, they can be chains, they can be colonies. But the key to this is most of these are going to do better in stagnant water. So what do you think the inside of a micro or macro aggregate looks like? Probably stagnant water. Cytoplankton, not zooplankton. There's many forms. Again, things just got dumped into these categories or these kingdoms because as the more we learn, the more we realize we don't know anything, or we only know a tiny, tiny fraction of what's going on in these huge communities that affect so much. And we're only looking at a snapshot in time. Like, it can change from one day to the next, and it does. So that's another thing to keep in mind, is that everything is in flux constantly. Diatoms got thrown into cytoplankton. Are they biology? Yeah, there's different forms of bacteria and larger forms of protozoa that are in this category. Are they a food? They are a food to larger animals in the food web. Are they a biostimulant? Sure, they're a great biostimulant. We know for sure that they add, again, a diverse set of nutrients into the soil system. So if they die, they're being mined by other bacteria or protozoa for their cytoplasm. And that in turn turns into enzymes. And as these protozoa who are low nitrogen, low end seed end ratio, they spit off a lot of nitrogen because they can't take it. And green seaweed. I mean, think about how big, how much biomass is in that system that's half underwater and half out of water during the course of a day. So you begin to see the diversity in this family group right here. And again, you're getting all of that in an AMO. Back to biofilm. So here's the graphic. Unfortunately, the didn't transition well over into this picture in the slide. But if you look on the left, you can see these tiny little spores. They set up shop, and then they start to multiply. And of course, they create the biofilm to stick them to surfaces. And then when we're building horizontal soil systems, I always tell people, yes, you want river rock, but you don't want it round. You have to crush it, smash it with a hammer, whatever you can do. You got to get rid of that polish so these guys can really take hold of that brand new exposed mineral. And that will allow them to build this biofilm a lot better. But as it goes through the process, you can see by the end of it, they've colonized, they've bred, and now they're releasing. And this is something else that they're putting more nutrients back into the soil system or the aquatic system. So yes, they're wonderful nutrient cyclers. So they're mining from the minerals. They're releasing back out into the water or into the soil system. All of these beautiful food sources for everything else to eat. Another interesting thing about biofilms is they do break down contaminants. So that's kind of a good thing, especially if you're in a soil system that has some issues with past pollution. A good couple of doses of a highly powerful AMO will help to break those nutrients or break those compounds down so that they're not as something that the plant can uptake, because we all know the cannabis plant is a very big accumulator and could test hot. if you are being forced to test. So what are the values of AMO? I think you're getting it between the fact that they're not present in soil systems, chances of you collecting them in a IMO box up on a mountain, probably not likely. In meadows and prairies, probably not likely because they just haven't been inoculated by the system as a whole anymore. So diatoms, I love to talk about these guys. There are so many beautiful shapes and sizes. They're just a huge integral part of the whole world. The diatomaceous earth from the deserts in the Sahara are blown into the upper atmosphere and in turn fertilize the Amazon rainforest. And there was an article done by NASA in 2011. And more recently, another one done, I think it was in 2018, saying that it was more than just phosphorus. The original one was like they just provide phosphorus, but now we're understanding they're providing a lot of other really critical minerals and nutrients to the Amazon River basin. So why do they matter? Well, the interesting thing about these guys is they can harvest light. All right, so if they're in the surface of the water, they're harvesting the spectrums we all see. But as they sink down and get into mud, they're harvesting another form, probably UV or maybe even a spectrum we don't completely understand yet. I have my friend Keith, which I'll talk about a little bit more, built a stainless steel collection system for me so that I could collect my AMOs from him. And so when he built it, he didn't really tell me. And I got down there and I figured it would be an open top, and it wasn't. It was completely enclosed. There was no light getting in there. There was only water being pumped in. And as it overflowed, it went into a sand filter that he put in the back of his property. So other than that, there was only aeration in there. And to my surprise, when I did my first collection, I microscoped it and I had thriving communities of these diatoms, and they were still alive and breeding because I could see smaller ones and bigger ones of the same size, same shape, not same size. So they were breeding and growing in a light-free or a completely void of light environment. So there's got to be more to them than meets the eye. I'd love you to look up the urea cycle and also understand ATP, which is the energy currency of the plants. Chip and I'll get into that a little bit more tomorrow, and so that you understand how, again, these things function. They produce oxygen in the soil system or in the water column. They're also an amazing source of silica. They have an incredible diversity. Again, we have so few studies on them. There's been some cool studies from NASA showing plumes of them as the glaciers melt and bring down all that wonderful mineral nutrient water out into the ocean. And again, to see diatoms in a aquaculture system is very unusual. The only place I've ever seen it was at Keith's farm, and I believe it's due to those incredibly nutrient mineral water that is bubbling up all around him. So how do I get them? Well, they're not easy to get. This was the only place I've ever been able to find them. And I'm going to introduce you guys to him or give you his contact information at the end of this lecture. Unfortunately, they are still in permitting in a number of states. Although crazy enough, California, which is the hardest one, he can actually ship out here as experimental. And so we've been doing that periodically as I need more materials. Pyrethrum. This is really interesting. I mean, this is like an entire ecosystem in itself. It has algae, it has fungi, it has microbes, has plant material. It's a food source. A lot of things actually live off this. We talked about zooplankton as the larger, they're almost like crustaceans and crabs, mollusks. They're not mollusks. I forget the name of it, but anyway, they'll live off of this, and it's so rich and diverse in food sources, kind of like you guys use kelp. It's just got all these micronutrients as well as plant hormones, and just a really beneficial material for healthy water cleaning, water quality. You might look at this and go, oh, that's algae, that's gross. Well, no, that's a sign of a really well-functioning aquatic ecosystem. And of course, you're going to get some of these. All right, so let's talk about bioenergy. Back to ATP. So the plant takes in the sunlight through enzymatic processes, creates ATP, which is used to move intercellular information. It helps to provide the processes of making exudates. It's just super value. So if you can save some of that energy so that you're not putting all of its energy into moving exudates down, water up, minerals up, you're providing a more nutrient-dense foliar spray to the plant, well, you're saving that currency. So that means that there's more food to do other processes. There's more fuel to move, the exudates and to put on biomass, and of course, more potency. So again, at the end of the day, this is the goal: is to get you inoculants that survive and provide multi-leveled benefit to your plant, and also again that diversity. In individual, you start looking at individual communities doing individual things. Each one is responsible for a specific task, but in community, they will all work together. So a community, a diverse community can be functioning, doing the same thing or diverse things. And so again, it's all about this diversity. And again, let's try to save some energy here so that the plant can put on more biomass and produce more of those critical secondary metabolites that everybody's looking for in a cannabis plant. All right, fourth phase of water. I touched on this the last time, and I've texted out a bunch of people about, look at this, think about this. It's really interesting dynamic that we have known about, I guess, for about, less than 20 years anyway. But what happens in this is we actually create H3O2. And I know you probably go, well, what the hell does that matter? Well, when that happens, you can see the little slide showing you the way these atoms are lining up. So the hydrogen atom is the red dot. Or no, the oxygen is the red dot. The hydrogen is the little bar, the bond. And so when they stack up like this, they actually push out any toxins, any minerals, or anything else, and it becomes negatively charged. And what we've found or seen in hydrophilic materials is that just it happens naturally that you have this easy zone, exclusion zone, and all of that water is negatively charged, and the body of water is positively charged. And this gets really interesting when you start thinking about the heart. So the human heart would have to be as big as our body to actually move our blood through our vessels. The blood itself is bigger in diameter than the vessel's internal inside diameter. So how does the blood get through there? Well, the theory is that we know biology, bacteria all have easy water inside them. That's how they use communications. And that's also how they attract nutrients. And it gives them the ability to communicate on a level because there's no resistance. There's no contaminants. And remember, water is the biggest acid on the planet. Everything attaches to it. So this exclusion zone is really interesting. And in a aquatic microorganism or aquatic environment where you have really, really dark, thick chocolate water, that's basically suspended humates and humuses. And we know those are hydrophilic, so they're going to have an exclusion zone around each one of them. So again, a superfood for biology. The interesting thing was they also did some experiments on UV. So UV light actually expands out the easy zone. And again, I'm not sure why that is. Hopefully there'll be some new work coming out to better enlighten us on this. But it's kind of an interesting thing, because in the soil, you're not going to have regular sunlight. You're going to have these far blues and far reds. And I found an interesting paper recently about how these other UV light forms are actually interacting with roots in the plant. And it's the opposite of what water is. In water, you lose your reds within the first 10 feet and your blues, I think, at 25 or 30 feet. Well, it's the opposite in soil. The far red penetrates deeper than the far blue. But again, there are all these potential energy sources that can be captured if you have very thermal or a lot of different easy water pockets throughout your soil system. You're increasing everything from your ability to hold nutrients and also exchange currents as far as positive and negative currents. Again, and that's what happens with when you're attracting nutrients to different sites. So I'm kind of theorizing that this is going to help increase your CEC, which is your cation exchange capacity. And I believe this has redox potential as well. I think that this is going to prove to be a real game changer in understanding some of these deeper scientific thoughts and processes that we're not going to go into today, but as more and more people start down these rabbit holes, it's going to be more and more people trying to research this and more information coming out, which unfortunately the COVID just stopped so much research because they're only allowed to have one person in the lab at a time instead of 20 to 50. So we've had quite a log jam on information, which is a bummer, but hopefully they get back to normal soon. So again, this is going to help increase biomass because the more easy water you have around, obviously the more cells can grow out or bacteria can grow out because it's more available. And again, the more biology you have, the more biofilm. So it's upping your game to another level. Health of our waterways. I know this is a sad picture, and growing up, I remember the picture or the commercial with the Indian with a tear running down his cheek, looking at a waterway similar to this. So when you're looking at trying to find these, aquatic microorganisms, this isn't a place you can go. And this typically is a picture of so many of the waterways that I see and study. Because it's the human expansion. We've just gotten so many pollutants into everything, and every time it rains, more and more wash in, and then they get collected and clog the system, the flow of the entire system up, so it gets backed up. You find microplastics, major plastics. You have everything in there, and so obviously, probably not a good place to collect AMOs. There's been some recent work in understanding tires. So rubber is expensive to break down and turn into tires. And it's also difficult to grow rubber trees. They're not really fast. So the tire industry looked into introducing microplastics to their tires. And now it's become apparent that is one of the biggest contributors to microplastics in waterways, because as the tire wears down, all of those little tiny pieces of plastic as well as the rubber come off. The worst part about this is, beyond the microplastics, is that the chemical they use to bond the plastic to the rubber is a fishicide. So what they've noticed out west here is, in some of these streams where all the steelhead and steelhead and sockeye migrate inland to lay their eggs, they die off because after a rainstorm, all of that bonding agent is washed into the waterways, and so they see large fish kills after rain events, and they finally tied it back into microplastics, tires, and bonds. The other problem you have with the waterways nowadays is the hydrocarbons. There's a tremendous amount of oil, gas, grease that are getting washed into these systems, and the diversity of these systems is collapsing. Now, you do get nutrients because we have the runoff from all the fertilizers, which is growing out algae, which again algae is not bad, but in balance, it's fine, but not when it gets out of balance. It just steals all of the oxygen. The more nutrients go in, the more it plumes, and then it affects the fish and the amphibians. So it's problematic. And so again, not the best place to be collecting an AMO. So how do we wildcraft AMOs? The same as IMO? Well, I had that conversation with Chris, and he agrees. He goes, yeah, as long as I showed you on the right here, this is the riparian system. The little chart with the blue shows how the water goes up and down. So like a high tide, low tide. And so you want to be collecting in the zone that is dry in between rainstorms. So a perfect world is collect in spring or fall, but a week or two after a rain event, so that the water level has gone down. So it's not mud. You can't put this rice collection box in soup. It has to be in water or in soil that the water has moved through it. It doesn't have to be bone dry, but it can't be mud. And then follow the protocol to installing an IMO collection box, and make sure that you get back there before the next rain. So it's probably important that you look at the long-term weather forecast. I mean, you don't want you to, take all the time and energy to do this, and then, 5 days in, when you're about to go collect it, you have a rain event. And because remember that creek, whether it fills up or not, is still going to be having water coming in from all sides. So therefore your box is going to get drowned. And that's not going to work for you. That's definitely going to turn it into slime. So there's a way to do this. And then let's think about the different locations of what we call riparian systems or drainage. So drainage is where you have mountains coming together and you have a valley that's all washed out. That would be considered a drainage area. Would it be a good place to collect an IMO or an AMO? I doubt it. I mean, you might get some there, but for the most part, drainage is just the water's moving through there very fast. So after drainage, it comes into watersheds. So watersheds are areas that are now they're a little probably flatter. The water's not moving as fast. We'll have time to collect and pocket. Perhaps could grow out a few of the organisms, but again, not as diverse as some of the lower places. So next we have waterways, which are like creeks and streams, where you have, again, during dry periods, they go completely dry, where, a stream tends to have water in it yearly, where waterways probably don't. They go like canals. They go through phases where in the wet seasons they're always water, and then in the dry seasons they pretty much dry out. Rivers are great for collections because you're going to have, again, diversity. So not just fish, but other animals as well as hopefully the microorganisms. Rivers are probably going to be your most beneficial. Lakes are going to be tricky because, again, for the most part, the water stays at the same level. Unless they're dammed and they drain them down in the winter, then that might be a place to go once they've drained it down, collected before it completely dries out. But I personally think the best place to wildcraft AMO is a healthy riparian system. A riparian system is basically a system that should be broken up into multiple phases, but it's from a certain depth in water to a certain distance away from the high water mark. And in that diverse system, you're going to have a lot of those organisms being transported by the wildlife that's around it. So you're going to have diverse plant life. So you're going to have all kinds of terrestrial microbes, but you're also going to have the aquatic microbes because of, the critters coming in and out of the water, the old watering hole. And so that would be a place that I would recommend. Now, again, you have to make sure you understand where that water source is coming from. If it's downstream from agricultural or industrial, probably not a good place to go. And you should be able to determine how healthy it is by the diversity of the animals that are there. So looking for scat, looking for tracks, microscoping the water is a great way of determining that. That the aquatic food web is probably a lot. Well, I wouldn't say a lot more. It's definitely as sensitive as terrestrial food webs, because as soon as you expose them to these fungicides, or any, for that matter, any side, you're going to have issues. You're going to take out diversity. So again, if you're going to wildcraft this stuff, you got to be really responsible and think it through. Use your common sense. Here's another source of ammo: aquaponics system. So there are more and more aquaponics systems popping up. One of the biggest things that is missing from these systems is the solids separator. That was the first thing I did with my experimental system, was to get a really good solid separator. And in the upcoming, I think it's April event, or no, maybe it's March, we're going to get into different types of solid separators. And if you got an aquaponics guy and he does not have a solid separator, or if he has one of those AES, which is aquatic ecosystems, sponge container, that's a great place to collect AMOs. It's kind of a pain in the neck. You got to pull out these giant sponges, kind of like what you saw in the back of a piggyback fish filter, where you have that little sponge that sits on top of the activated carbon bag. That's basically been duplicated and scaled up for some of these aquaponics systems. I put Nelson Pate up in here because if any of you out there are interested in getting an starting an aquaponics facility, please do not use these guys. They are aqua shysters. They have done so much damage to the industry, overselling you things, not explaining it properly, and just selling stuff that flat out doesn't work. My recommendations is you look to the aquaculture industry. There's plenty of information online on how to build these things at home cost effectively and not paying 10 times the price of a plumbing piece that you can go to the hardware store and get. So the nice side of aquaculture too is that if you can find an aquaculture system near you on the shore, on the coast, they're all going to be salt water, so not a good place to collect your AMO. But as you go inland, look for trout farms. There's a bunch of farms in the Midwest that are coming up, especially around the Great Lakes. And those are all going to be freshwater species. You want to ask them if they have drum filters. That's going to be the source of collection, because a lot of these guys are not going to have a collection tank post or downstream from their drum filter. They're just going to dump that stuff out. And that's the goodies right there. That's all of the fish manure, as well as all of the saprophytes that are breaking down the plant matter, the feed, everything else that's going to be collected in that. The other thing is a fish tank. Like I tell people, there's no cannabis grower that shouldn't have at least a 55-gallon fish tank. It's not that hard to take care of, but what you can get out of it is you can actually get these AMOs from the filter system. There's a couple different types of filter. Don't get it under gravel. I don't think they even sell them anymore. The canister filters are fine. This little filter I popped up here is a really cool little device. It works great. It's air driven, so it bubbles the air up and pulls the water around it into it and then collects it. So it's even easier to do this way, and then just have this as a supplemental filter, not connected to your main filter. But this is a great place to get AMOs. And then fish feed. So I've had people say, oh, well, there's GMOs, there's all this stuff in fish feed. We have to understand that once that fish has consumed it, its gut biome has broken it down. It's no longer going to transfer from one bacteria to another. So the GMO has only affected the plant itself, and it cannot be transferred biologically from the plant to the bacteria. So please don't stress on that. You're not going to do anything wrong. But here, there are a couple different sources of AMO. Stabilizing AMO. So you can do it yourself at home. I showed you this really cool whirlpool. So this is one way of getting oxygen into a water column, but not the best. So let's reverse this. Let's have a column of bubbles going up instead of the air coming down. And in a cone bottom system, you're going to get incredible mixing because the air is coming up the middle. It's pushing all the liquids down the sides back to the center where they get hit by the air and drawn back up again. So that's by far the best way to bring DO into increase the dissolved oxygen in the water column, which is the most important part of stabilizing aquatic microorganisms. For the most part, they're going to be anaerobic, so therefore they're going to be smelly. And you want to aerate these as long as you have to start getting a sweet smell. Again, I wish everybody had a microscope and knew how to use it, but I know a lot of people don't and don't have the time to invest in learning a whole new skill. So if you get it to smell sweet, you're in the right place. And my recommendation is just if it's starting to smell sweet, let it go another 24 hours. So you're really guaranteeing that you're getting rid of any potential pathogens in that system. Speaking of pathogens, it's quite often that aquatic water, aquaponic water, will test false positive for E. coli. There's a lot of different E. coli kind of or like biology, or bacteria, but unfortunately, the way we've looked at E. coli is that if it's got E. coli, it's got a problem. So a lot of times they will tell you if you have an aquaponic system, you got to wash the heck out of the lettuce. You can't let your fingers come in contact with the water that it's grown in. You got to be really careful about getting the roots off. It's just all kinds of unfortunate regulations that are driving that industry. And this is part of a soil or a healthy water system. It's a false positive. So that's the goal with aerating, is you're going to get rid of any of these pathogenic or anaerobic organisms. Agitation is critical, and that's why I say a cone bottom with the water or the air coming in at the cone, the bottom of the cone, is going to be by far the best and easiest way to stabilize this stuff. Do not use totes, because totes, what's going to happen is you're going to have all of this settling out in the corners and at the bottom, and you can't get to it. And even if you move the stone over there, the solids are going to settle on the opposite side of the tank. So not an ideal way to try to stabilize these, especially if they have solids. Now, if you've already taken the solids out and it's just water, totes will be fine. You're just trying to increase the dissolved oxygen in the water column to kill off any pathogens. I talk about the microscope again. I know you guys are probably not all going to rush out and get a microscope, so just use your nose. As Chris said, I loved it. Your nose knows. It's probably one of the most valuable defense mechanisms the human body has in the regard as detecting whether something is good or bad. Applying AMOs. So again, you have a nice sweet smell to it. Foliar is amazing. When I apply the products that I make to plants, I notice instantly within minutes, and sometimes 24 hours, that you will see this incredible color change in the plants that are treated. They just get more vibrant in their color, deeper colors, and their luster changes from foggy, hazy to like crystal clear. It's just a huge difference that is visible, especially if you pay attention. Soil drench, great. Now you're getting it down into the soil system with the hopes that it is colonizing and not just becoming a biostimulant, so inoculating. AMOs are great for chips. Like if you soak chips in them, those organisms will soak up inside the wood and hopefully colonize again, or most of them will colonize. And that's a great way to make compost, is to spray it with an inoculant to get it going. And again, even if these all don't stay, they're going to be foods for the ones that do. And I guarantee you are going to get flagellates and amoebas. And that's for sure. And those are nutrient cyclers. So they're the key to the whole nutrient cycling system. If you just have bacteria dominant soil and no protozoa, you're not getting that nitrogen transfer. You're not getting that nutrients that you need. So for an AMO to be applied to a compost, like Commercial compost, fantastic, because commercial compost was cooked to 160 degrees for 5 days straight, turned 5 times, whatever. Every state has a different regulation, but for the most part, it's going to take months for that, or if not years, for that for those predatory organisms to come back and start functioning again. And again, it's going to take a long time for them to build up. So if you inoculate compost with this stuff, you're out of the gate going to be fantastic. I've done it to the point where built the compost pile, inoculated it, and then 4 weeks later had very, very biologically diverse and intense compost. So it's a quick way of making something that is void of, for the most part, other than bacteria life, into something that's functioning fantastic. Oh. No, all right. Benefits of AMO. All right, so this was a study done in Australia recently, Australia and New Zealand. And these guys went out and collected 400,000 species of marine, terrestrial fungi, endophytic bacteria, marine bacteria, actinomyces, and rare actinomyces and terrestrial actinomyces. So it's the best example I've found of both a bacterial and a fungal overlay of what is found in these diverse soil systems. So they went everywhere from prairies to mountaintops to rainforests to deserts to different stages of climate change from the bottom of the mountain to the mid mountain to the top mountain. And again, as much diversity as they possibly could find. And they collected 4,000 species. And the interesting thing is that actinomyces are considered pathogens. They're considered problematic. And 3/4 of this pie chart is actinomyces. So, is this a real big issue that you're potentially could be getting all these things into your AMO? No, it isn't. Because if you look, the marine version of actinomyces is very small. And the beneficial part of the chart is there's no actinomyces at all. So the real bad actinomyces are already in the soil. And this might be a way, again, to counteract that by putting more good guys in play. And I would have loved to seen this study being done perhaps 2,000 years ago before the Great Expansion. Perhaps that pie chart would be completely reversed. In other words, way more good guys than bad guys because of all of the impact that we've had on this planet over the course of 2,000 years. So think about that. Again, all of these guys are going to create biofilm, which is critical for aggregation. And the key to soil health is aggregation. So it allows, again, gas exchange, oxygen down, CO2 up, water infiltration. Of course, your compost tea is going to get in there better. So it's all about getting that biofilm and getting that aggregation, which means high levels of microbial activity. Is it a biostimulant? Yeah, sure it is. Not all of those organisms are going to survive. So therefore, they are going to be a biostimulant. So it's basically feeding bacteria or feeding protozoa bacteria and diverse bacteria with different new nutrients stored in their pantry. So the understanding is that the bacteria mines things, and inside that bacteria has a pantry where it collects all these things so that if something happens, say a pH change or oxygen level goes down or up, they can pull things from their pantry to change their internal chemical makeup. And so when the protozoa eats them, all of those are released, plant available, into the soil system, and usually right around the rhizosphere. So think about giving your plant some things that it probably doesn't have in your soil system, some rare earths or some micronutrients, because there's a tremendous amount of micronutrients circulating in aquacultures systems. Chris talks about using fermented ocean water because of all the micronutrients. So the same idea, these aquatic bacteria are going to have micronutrients that are probably not present or at least in diversity, and by far not plant available, because even if you have the trace minerals in the soil, doesn't mean that the roots got to them. So this is a great way to get those trace minerals in contact with the rhizosphere and break them out plant available. So is this the missing link? I truly believe it is. And, I think that now that you're aware of it, you start thinking about it. Common sense would say yes, because we don't have these mass migrations anymore, that we are not getting a regular inoculation of these AMOs. And again, can they live in soil? Yes, some of them can and do. That I am positive. Just flagellates and amoebas. You're going to see tons of them in this AMO. And if you're in a good, healthy soil system, you're going to see a lot of them there as well. And some of these other bacteria, like phosphate solubilizing bacteria, absolutely they live in the soil. And I'm definitely going to be diving down the rabbit hole of getting hopefully some more information on some of these organisms that can live in water and in soil, so that I can better the presentation and other people and the understanding of just how valuable these kinds of inoculants are. So this is the contact information for Fish Brew. They do not have this product available or licensed in every state yet. They've been working diligently on this for 3 or 4 months. Unfortunately, they didn't think it was going to be this complicated, because originally, as long as you just say soil conditioner, you have no issues whatsoever with selling this in any state. But the states have gotten a little bit tighter. Look, everybody's looking for money, so they all want them to permit. And so they're in the process now of getting these permits across the country, so it doesn't matter where you are. But I figured I'd give you the contact information for now, so that you could log in. You can check out the website. It's just a landing page, but you have an email now, so you can actually start reaching out and letting them know where you are. And then they could focus on getting that state licensed as quick as possible, so that they get the product to you. I don't believe it's going to take months and months to get through this. I think they'll probably be able to have pretty much the vast majority of states up and running by springtime, so just in time for your outdoor grows. I've already got some big ag companies who are just can't wait to get their hands on this stuff, to start putting some diversity back into their soil systems. When you get into large field production, it's difficult. I mean, they have giant spray equipment, but they don't have affordable products. And Keith has promised me that he's going to keep this product very affordable. The numbers haven't come out yet. They're working on that as well. The biggest issue to this product is shipping, because it's coming in liquid. But I can guarantee you this: the water is high nutrient value. Just the fact that these fish are so healthy. I pray to God someday he actually does farm tours, because it's just one of the coolest things you can imagine, where you're walking around with just fish everywhere on these raceways, and the health of them, the colors that come off of them. If you've ever had an aquarium, you'll know what I'm talking about. And he's by far, there's no synthetics whatsoever used on the property. The feed's as clean as they possibly can get. They don't use any hormones. They don't use any antibiotics. And I've known Keith for a long time. He and I crossed paths, I believe, at a aquaculture system 12 years ago, 13 years ago. And we've stayed in touch because, he's very much a geek like me. At first, he actually would laugh at me. He's like, I know there's value to this, but seriously, why do you want to play with this stuff? And, I'd poke back at him, but I knew there was something here. And it has been a cornerstone of all of my products since that first day, when I brought a vial of some stabilized fish shit down to Elaine. I didn't know who Elaine was. And I got in there and I said, look, Jeff Frank told me I had to see you. He said that I need you to tell me what's in here. Because I put it on plants and it works great, but I don't know why the plants are reacting the way they're reacting. And so she took a drop, put it under a microscope slide, looked for like I don't know, 5 seconds and just picked her head up and said, "What the hell is this? Where did you get this?" And she ran on to rant about how she'd studied, manures all over the country, all over the world, all different types of animals, and had never seen anything like this. And I said, "Well, it's fish frass, fish poop." And she's like, "Well, how the hell do you get fish poop? You can't go behind it with a pooper scooper." We had a good laugh. And I explained to her, I just invented this fish filtration system that allowed the suspended solids to settle out before getting pushed back into the aquaculture tank. And all I have to do is just, once a week, open the bottom, pour it out, and I stabilize it until it starts to smell sweet. And then, then I know it's going to have incredible plant reaction. So in many ways, this is a great resource for you guys. I know the benefits of it are multi-tiered. Again, whether it's just a stimulant or an inoculant. And it looks like I went way too fast through that presentation. So let's hit some questions here, and I can always go back and pull up the slide that the question's related to.
All right. Thanks, Leighton.
Yeah, I did. This was a 2-hour presentation, but I did not want to go over the time and not give these a chance to ask questions.
Yeah, I think that's good. I think people have a lot of questions. So let's start back at the beginning here. So Dylan was asking, what do you think the blue lab that Aquaponics Steve started? What do you think of the blue lab that Aquaponics Steve started doing? You know who he's talking about?
That would be Growing with Fishes, Steve Reisner.
Not sure. Blue Lab is the name. Anyway, he said under a scope. It's pretty cool.
There's a couple companies out there that are doing this. I think the first one I came across was Fish Shit. And when they originally had the product, you'd open it and it would gas off hydrogen sulfide really bad. So it stunk. So right away I was like, I'm not so sure about this. And so I did put it under a microscope. There was no flagellates or amoebas. It was just bacteria. I mean, it's better than nothing because they are aquatic bacteria. So you're going to get some phosphate solubilizing, some nitrifiers, and some potassium solubilizer. But you're not going to get the predators, and the predators are where it's really at. That's how to get that cycling system up and functioning as fast as possible. So throw the question back at him. Since you microscoped it, what was the level of amoeba and flagellates? Were there any yeasts? Were there any rotifers or stalk ciliates? Was there anything other than bacteria?
He said yes, it is Steve Reisner, and he said it looked pretty cool under the microscope. He said I think it turns blue because of cyanobacteria, but he's not really sure.
That's interesting. Well, funny enough, I have the deepest, most respect for Steve Reisner. I think he has an amazing knowledge and overview of this. I know he studied under Sylvia Bernstein, who was, another, what I want to call forefather of this. And again, so he really dove down the rabbit hole. I think he came out of fish tank maintenance, if my memory serves are correct. And so when he found out about aquaponics, he was like, wow, that's really cool. So it was like an easy leap for him because he already understood aquatic chemistry, which is the hardest thing. So whenever somebody asks me like, oh, I want to start an aquaponics facility, I'm like, all right, well, what size? And have you grown plants? And have you grown fish? Well, I've grown plants. Okay. Fish is a totally different animal. Now you have to be good in fish and plants. That's exactly why I did what I did. Sorry, I was supposed to put that up. Was create Kingdom-Aquaponics so that I could actually bring the benefits of the aquaponics or the aquaculture to you. So it's still aquaponics because I'm giving you the things that make the plant grow. And so if Steve has a product out there, my gut feeling is it's solid, and I would encourage you to purchase it by all means.
All right. Let's see. When heating a bed with underground radiant tubing, which layer of the soil profile do you recommend running the tubing in?
I would recommend it in the E horizon down at the bottom. Now, we're going to get into soil systems a little bit more in tomorrow's session. These horizontal soil systems, as I call them. But I want people to understand is that the benefit of having that E horizon is that you have a place for excess moisture to go. The key is you have to have the ability to see if it's full of water, or if you're flooding it, because you do not want the water table to get all the way up into the A horizon. The water should never exceed the top of the E horizon. So what I usually suggest to people, if they can, is to either put a pipe down in the corner where they could take a stick and dip it down in there, like an oil dipstick, to see and pull it back up and see what the depth of the water is, or do like a container with a plate or saucer around the outside edge. Now that plate or catch tray should be the height of the E horizon. So in other words, when the water starts dripping over the side of that plate or catch, you've hit the height of the E horizon. That's the key to the whole thing. And to store water in the E horizon is no problem whatsoever. It's not going to go anaerobic because you have the sand that's protecting the fines of organic matter from getting down into it, which would go anaerobic. So as long as you don't have organic matter in that water, you're not going to have a problem. It's going to be anoxic, but it's not going to be anaerobic. And so anaerobic would be void of all oxygen, and anoxic would be from about 6 parts per million down to 1 part per million. And of course, you're not going to keep watering it until you notice that E horizon is almost dried out. Then that tells you it's time to start watering again. Now, next session, we're going to get into crop steering using water, which is, I've heard of 3 people doing it so far. Brandon Russ is not using water to crop steer or epigenetically crop steer. He's using nutrients, but this is going to be another tech that these big grows are going to freak out about, because it's really taking this biological soil system, connecting it to the aquatic or water influence, and allowing the plant to go through certain phases and processes without totally stressing out. Therefore, again, creating the highest level of secondary metabolite production that you can possibly achieve. So hitting full plant potential.
All right. Next question is, if you irrigate your living bed with aquaponics, are you going to fundamentally change the rhizosphere? Okay,
the rhizosphere is the coating of the root. So that's rhizo meaning root and sphere meaning coating. So by adding aquaponic water, you're never going to affect the rhizosphere, but you are going to be introducing specifically aquatic bacteria. I don't know if you're going to get fungi through there, because if you're using drip tape or Netafin or blue mat, you're going to have problems, because you're going to plug up your emitters and potentially have biofilm in there. So anytime I run into this situation where people want to use products like this on large scale, I say either you got to have an enzymatic cleaning after, so run an enzyme through there that's not going to harm the plant or the biology, or humic acid, something that will strip out biofilm, or blow air in it. If you blow air through it like we do back east in the irrigation pipes, that's gonna blow the water out of any little dips or rises in the pipe. Because if you don't blow it out, those little dips and rises are definitely going to go anaerobic on you, and then you're going to go backwards. I think Chris made it clear that once you get aerobic, and if he didn't make it clear enough, I'm going to pound it home. Once you get aerobic, you do not want to be applying anaerobics back on it, because what you're going to do is you're going to step backwards. You're going to take the soil backward in succession, not forward. Can you use a little bit of anaerobics? Sure. You have bokashi, some of these ferments, no problem. But you're not going to want to irrigate your system with an anaerobic. So that would be like a soil drench. That would be a big no-no. You create a lot of problems.
All right. So you had mentioned using humic acid possibly to break down chloramine in city water. Can you elaborate on that a little bit more and what people could do there?
Yeah, absolutely. So I would probably recommend, if you're going to buy something off the shelf, buy BioAg. Dr. Faust has done an incredible amount of work over the years, both abroad in Russia as well here in America, finding exact pockets of lenidite, which is brown coal, and mines it. And I'm pretty sure that he is the only one that's not using acids and bases. I believe he uses biology to break it down. And basically what that is old plant matter. So the other source for this is if you have really good compost, you can make a compost slurry. So that's like a pile of compost, fill it with water, maybe just a little bit over the top of the compost, and then agitate the heck out of it. Whether you put a plaster mixer or paint mixer on the end of a drill and beat the hell out of it for 10, 15 minutes, or you just start, agitate it or use your hands to knead it, and then drain off that water, which is no longer water. It's going to be humic acid. That's a great way to do it yourself at home. And, again, you're going to probably get some good guys out of that compost if it's biologically active. So you're going to be getting a double win. You're going to get the organic matter, which is critical in breaking that covalent or, ionic bond. The organic matter is what does it. And so this is a little not so fun fact, but the vast majority of water treatment facilities in America fail on a regular basis because they're pulling their water from outside sources. So reservoirs, canals, what is the other one? Brain cramp. Basically a way to move water. The Romans did it.
Aqueduct?
Aqueduct. Thank you. So in exposing the water to the air, the chances of you getting leaves and other organic matter into that water column are huge. So whenever an organic matter comes in contact with a chloramine, it breaks the bond. So now you're not getting that level of antibacteria or antimicrobial that you're looking for. So by default, the water companies will constantly increase the combination, and they have two choices. They can either add chlorine and then add ammonia until they see the ammonia spike, which means that all of the ammonia is now bonded to one of the chlorines. So that the only time they'll see ammonia spike is when everything's bonded. Or they do the opposite. They add the ammonia and then put the chlorine in until all of a sudden they start seeing chlorine building up. But the problem is they're constantly adding more and more and more into the system, figuring that they're going to have some organic matter tied up in the system. And if they get too much of that, they start to grow out, or they start to create these chemicals, kind of like VOX, volatile organic compounds, and those can be toxic. Those can cause health problems. So they're tap dancing on this really delicate balance between having the water be stored outside instead of in a water tower and a pipe, getting that organic influence, trying to maintain the bio side, so that they're not getting E. coli spikes. I mean, we've all had towns that had to flush their water system because they had E. coli spikes. So I'm glad I don't work with those guys. I did do some work early on with helping them to understand what their nutrient loads are doing downstream, so that they could decrease it. This was when they switched from measuring nitrogen to measuring phosphorus, and then they just took nitrogen right off the table, which was insane because they're both causing problems in the waterway. I did some work with one of them, and they gave me a tour. They explained all this to me, and I was like, man, how complicated is this? And how everything's changing constantly on you. How else do you stop that? And the only way they can do it is just keep adding more and more and more till something raises. And then stop, and then test, and then add more and more and more. So that's what I'm saying. The high level of humic acid is going to break that bond permanently. The ammonia will be available for the biology to uptake, and the chlorine will gas off if you're aerating it or stirring it, and you'll be all set.
Awesome. All right, thanks. So someone is commenting that they use micronized silica in shrimp and some fish ponds to promote algae and diatoms, which make it healthier. So could you transfer this method to the soil?
Yeah, that's basically what I'm saying. But the good news is if you have diatoms, you have silica. So they're naturally going to go through a system. I mean, the loop that NASA did was amazing. I think it was over like a 6-week period, and they did a time lapse, and you can actually see the plume of these diatoms growing out away from the shore as the glaciers were full melting. And then as the glaciers stopped melting and stopped producing that high mineral water, the diatoms, the color changed, and the diatoms shrunk back down and fell to the bottom of the sea. So they have a relatively short life cycle. So as long as you have them in play, they're producing the silica and making more. So you're going to have everything you possibly need.
Great. Would collecting water from your local rivers and add... that doesn't seem like a complete question, but would collecting water from your local rivers and add it to your feeding tanks? I guess the question is, would you do that?
Oh, yeah. Here's a great one. And this was part of what I cut out. So back, must be 8 years ago now, a hydroseeder came up to me and he was like, look, I got a problem. I have this client, they want, everything to be organic. And so I can't spray my regular seed with the green coating that has the organic or not organic, the inorganic Miracle-Gro coating on it. And so I can't use my regular seed mixes. So I come up with a paper product to support the seeds. It was kind of like a pit moss. It might have actually been pit moss, but in its early days. And he wanted me to help him with some biology, in which I said, yeah, it's no problem. I can give you jugs of it, and you just dump it in as you mix your seed and your mulch together. And I go, but what would be really good is if you went to that river and pumped your tank full of river water, because that's just going to add another level of biostimulant to the whole system. So he did it, and he sprayed it, and then it was a really big job. So he had to spray at different sections. And so by the last section, he didn't have time to go to the river, and he used hydrant water, which is what they traditionally do. And basically what happened was the grass didn't come up as quick. It didn't have the same colors. It wasn't as thick. And he called me and he brought me over there. He goes, how do you explain this? And I'm like, well, the water killed the biology back because it was all chloramine, and the river water was loaded with more biostimulants. So the river water was a double plus, and the town water was a negative. That's why you're having such a big difference. And so he ended up having to go back and reseed it because the client complained that it just didn't look right. So it was kind of like I made a mistake making it too good.
Nice. Great. So another question, are diatoms paramagnetic, and microbes in general, are they paramagnetic?
All right. So I would say no, but I would say that organic matter is paramagnetic. So paramagnetic means very low voltage. And as I discussed about easy, each bacteria is filled with easy water. So it would technically be paramagnetic. But a diatom is, think of it as like a glass house. So it's inert. So it's not hydrophilic. So it doesn't hold water. It protects the cytoplasm on the inside of the diatom from mixing with the water on the outside of it.
Okay, great. Let's see. Let's see. So would natural wetlands be a good place to collect AMO?
Yes, I think a wetland would be like a swamp or a marsh, something where you would have influences. Like in the summer, it would be perhaps dry or a lot lower. And then, of course, in wet seasons, it'd be a lot higher. So that's where you're going to get those interactions that I was talking about, where during the wet part, you're going to have all of those AMOs all over the place. And as it dries back, they're either going to cyst or they're going to stay alive. And this is where, I had a wonderful conversation with James about the cycles of the endophytes and how I theorized, but I wasn't sure. I said, I theorized that if the leaf falls into the river, that the endophyte, in fact, ends up being a saprophyte, but an aquatic saprophyte, because I know there's tons of those. And he said, look, we're working on that now. That we do have some papers and some information, but it isn't it isn't well studied at this point in time. And again, we have so much work to do. And moving forward and understanding these relationships between communities and groups, and the fact that there is 3 cycles to a fungi, is mind bending alone. You guys just started learning about fungi, and then to say, oh God, it's got this endophytic stage, it's got a saprophytic stage, it's got an aquatic stage. It's a lot to fucking wrap your head around. So my hypothesis is, I'll stick my ass out the line here. I know for a fact that you're still going to have some of those biological components, whether they're rotifers, stalk ciliates, nitrifying bacteria, phosphate solubilizing bacteria, potassium solubilizing bacteria. I guarantee you're going to have all of those in that soil system adjacent to the water. After the water's gone down, they're still going to be present in that soil. I mean, I've seen these in soil systems that were anaerobic or were dysfunctional to some degree, but they had all the good nutrients, but the soil aggregates had collapsed. So they didn't have what I want to call good respiration or good water infiltration. So I saw them. I identified them. I took them out, harvested them, grew them out, applied them into a plant side by side, and it worked. So yes, riparian systems I love because of the wild diversity of animals, where in a swampy or marshy area, you're going to have less. You're going to have more reptilians, amphibians, potentially fish, hopefully. But you'll have even more diversity in a riparian system where you have that forest that's untouched, which is kind of where Chris is always suggesting to go for an IMO collection, where it has not been disturbed in a long time. Now, remember, most of America has been logged. So, it has been disturbed, but that was, 150, 200 years ago. So a lot of that has come back to some degree, but that's why I call this AMO or AM the missing link to what would have been an amazing IMO collection, because we don't have that migration anymore. If we have, if all these animals were running wild all over the place, you wouldn't have to go hunt for an IMO section. Wouldn't have to hunt for an undisturbed location. And in that undisturbed location is probably a pocket. So you might have some organisms or advanced organisms like animals, critters, but you're not going to have migrations running through there. So those migrations stopped, 300 years ago when we wiped out the buffalo and the geese. And so, again, it's like, my goal here is to get people the ability to turn dirt or engineered components into a living soil system as fast as possible. And by doing that, you're going to need inoculants. You're going to need products that actually work. And so, again, this has been my rabbit hole for the last 12 years plus. When I was a kid, I was doing it. Was playing with these AMOs or AMs, whatever you want to call them. Yeah. Swamp is a great idea, but again, you're looking for incredible diversity. So the more critters, organisms, both in the water as well as on the shore, and interact with that water system, the more diverse you're going to be able to collect those AMOs. But remember, if that box gets wet, into the compost pile because it's going to just turn to mush.
Yeah. So maybe can you clarify that a little bit more? So you're talking. I mean, are you still talking about using rice like you would with IMO and but just making sure it doesn't get saturated? No, you want it to just get
right. So the problem with rice is that it has to be cooked al dente. So a little bit on the dry undercooked side, so that the outside of the rice is soft, but the inside is still hard. So imagine if you took that al dente rice and got it wet. It's gonna basically just turn to muck because you still have all of the good fats, sugars, starches inside the center of that coating. It's there for the organism, not to come out in the water. So you follow me? So if you make wet rice, you're not going to get a good IMO collection. If you cook the rice so that you would eat it, you're not going to get that collection because the softness on the outside is only for the organism to stick on, but the real food that it's after is in the inside that has not been cooked yet. So if you overcook the rice, forget it. You're wasting your time. You're not going to get a good collection. If your undercooked al dente rice sits in water, you're going to have a problem because, sorry about that. You're going to have a problem because it's going to turn to mush, and therefore you're not going to have the food sources that those organisms, IMOs, are looking for.
So, and just to tie it all together, and this is a question. I think we've got the answer now. But so you're doing the collection with the box on the flood bank essentially, so that you're getting those aquatic organisms, not getting it wet, but then you're taking it back and reintroducing it to water and stabilizing it with oxygen, right?
No. Well, no. You would have to wash it. So, probably like a good cone bottom tank in agitation, some kind of agitation to get the organisms off. So in my process, when I make product, I use compost, worm castings, and AMO. And I put it in a system where I agitate it. I soft collide it. So basically, I'm pulling those organisms off the side of it. All right. So somehow you've got to detach those organisms that are stuck to that rice and living off of it. So you're basically trying to remove the biofilm. So it's going to be more than just aeration, I guess. I mean, my gut feeling is that if you left it in there long enough, probably the pieces would bang around enough to strip that outer coating off and release the organism. But my gut feeling is that might take a few days and a low, like kind of like a rolling boil, so that you didn't have so much water that the rice particulate didn't hit against each other. So that's why I'm almost thinking like a mechanical method would be better. So again, go back to like a compost mixer, or not a compost mixer, a compound mixer. It has two blades that are like opposite each other, and they're wavy. So when it spins, it's kind of pushing one up and pushing one down. So again, and on low speed, because you don't want to destroy these organisms. You just want to pull them off, softly pull them off. I call it soft colliding. So if we're underwater and I punch you, you're going to feel me. You're going to feel it, but it's not going to hurt. It's not like when you come above ground and I whack you, it's going to hurt. So the goal again is some kind of mechanical agitation to pull the organisms that have sunk their teeth into that carbohydrate, pull them off. And then once you get done, then you'd want to put them probably in a paint strainer and strain the rice out of it. So pour the whole bucket through a paint strainer. And that way you'll get the rice out of it, and then you'll just have the organisms.
Okay. I think that clarifies it. So somebody's asking about the natural sponge soaked in aerated carb water. Like this is a method, and they're saying so we should not do that.
All right, I say that one again. Let me see if I can.
They just mentioned natural sponge, the natural sponge method.
Oh, the natural sponge method. So that would be like you have a little fish filter on the back of your tank, right? So you're sucking out water out of the tank. It's going into the sponge, into the carbon underneath of it, and then recirculating back into the tank. So that sponge, you basically take that sponge out when it's dirty, put it in a little bucket with a little bit of water in it, and just wring it out. Just squeeze it till you get that thing clean, and then keep dipping it in clean water, and then squeeze it some more until that sponge is perfectly clear. And then at that point, now you've gotten all the organisms off of it. Like a lot of times, people just stick it underneath the sink and just keep wringing it until nothing comes out. Well, I'm trying to catch every single little one of those organisms. Now, the key to this is that you can actually grow them. If you have the organisms separated from like the rice or from the soil or whatever, and you put them in a cone bottom brewer, again, do not use a flat bottom brewer. And you add just a tiny bit of molasses, in 30 hours, you will have an incredible plume of protozoa, flagellates, and amoeba. If you go over the 30 hours, you're going to start losing them really quickly. If you go at 24 hours, you're not going to quite get to the point where they breed out, where you've given them enough time to multiply. So that's a great way of taking that little bit and turning it into so much more. And then once you get ready to pour that or apply that to your plants, that's when I would add a little bit of perhaps fish hydrolysate, maybe perhaps a little bit of humic acid, and maybe a little bit of plant hormone. But do not brew them with those. I've done extensive research and work with brewing with different types of hydrolysate, thinking like, oh, well, they come from fish, right? So it kind of made sense that I would start adding, hydrolysate. What's the other one? Chris forgot it too. There's three types. Emulsion. There's emission, emulsion, and hydrolysate. So hydrolysate is you use the entire fish. You do not strip the oils. Emulsion, you use heat to strip the fish. And so you've cooked all of the biology. And emission, you stole the fish oil, which is what you're really after in the first place. So I used emulsion, emission, and I used hydrolysate in different types, and again, side-by-side tests to see what the effect it would have on my growing out these predators. And every time after 30 hours, I would see a decrease in numbers as well as diversity. So for whatever reason, they just don't play well in a brewing type scenario. But I have had wonderful luck by adding them after I'm done brewing and then applying them. So I'm applying them with the food source, but not growing them in the food source.
Okay, great. That actually answers the next question. Someone was asking, will fish hydrolysate bring me AMO? So it sounds like the answer is not really.
Well, no, that's not altogether wrong. I mean, inside the gut of the fish is going to be all kinds of AMO. On the outside of the fish is going to be all kinds of AMO. And incidentally, let me go back a slide because this company is also going to have a fresh hydrolysate too. So they're going to have two kinds of tea. They're going to have a light and a dark. I believe they're coming up with all the names, and they'll have hydrolysate. So again, you could get all of your goodies at one place if you want real fish products. The problem with the market as a whole is, let's face it, everything's a profit driven. So a lot of these companies that are telling you it's hydrolysate, it's really not. It's an emission because they stole the oil, and the oil is the key. That's the one thing that you really want, because they use the oil to treat soy, to trick the fish into thinking that it's fish meal. Because the biggest problem with aquaculture is you have to feed fish to get them to grow. So if you don't have fish to feed them, then you have to cheat, and you have a couple choices. One of them is to soak some of these plant-based nutrient packs in that oil, so it tricks the fish to thinking that because it smells like another fish, they'll eat it. And then there's one other way, which is to harvest fish meal, take the leftover bits of the fish and grind those up or strip them, and then try to use that to to blend in with those plant bases. So again, if you want real hydrolysate, go to that company.
All right. Let's see. So somebody was asking. Oh, actually, we had a clarification here from Clay about the question. He was saying Steve at Potent Ponics. He gets a natural sponge, then aerate overnight in car water, then put it in a stream or lake to get it oxygenated, and then collect AMO that way.
Oh, that's genius. So he's just taking a natural sponge and putting it in a lake.
That's what it sounds like.
And letting it colonize naturally.
Yeah.
That's a great idea.
Yeah.
That's a great idea. No, like the kid's a genius. Cool. He's a really smart guy.
All right. Another question was, can you use AMO in the process of making the different successions of IMO? Can they be used together?
All right. So in making IMO, the goal is to get these terrestrial creatures, stabilize them in sugar, blend them with a carbohydrate source and a carbon source to grow them out, then introduce a undisturbed or least likely disturbed soil you could find to make IMO4. So now you have grown out this process to the point where you've introduced it to the soil. At that point, I would introduce the AMO, but not before. And then the IMO5 is when you introduce manure to the soil. So now you have another nitrogen source. So I would add it at 4 or 5, or you can always, like we did on the Colorado project, was he sprayed a liquid IMO3 on the E horizon. Then we put down the first layer of the A horizon. They applied an AMO to that. Then they applied the second layer of A horizon, applied an IMO to that, and I believe they an AMO to that, and then an IMO4 to that, and then they added the top organic horizon and added and applied another AMO, a liquid IMO3, and top dressed with an IMO4. So it was really a all out inoculant. And when I say inoculant, when we sprayed those layers of soil, it was just till they were glossy. It wasn't like we drenched them. You don't need to drench it. It's just about getting a nice glossy color to the soil, so it's not dry, but it's not like muddy. It's just glossy. So you're not using a ton of it, but that way you've inoculated every single layer, and that's how you get to that point where you have a soil system that punches it out of the park on the first run. That was something I learned in landscaping and how to, on scale install, fields of engineered soil. And it was like, well, if we want to accomplish the fastest transition so that it looks like it's always been here, then we want to apply between every 6 inches. So no more than 8 inches deep without an application. And it worked. I mean, it was unbelievable. Like, I think I've told the story enough times that the architects were beside themselves that there was no plant loss. They expect 20% plant loss on these jobs. And I had 0. And it was because of these methods. And this was a trashed site. There was just, when we were digging, for all of the driveway, the underground services, the septic tanks, the holding tanks, the infiltration tanks, all of these things, we were finding wheelbarrows, paint cans, bricks. I mean, it was just, people just dump shit on this property forever. And so I was in charge of bringing in, all of the soil. I think we used over 1,000 yards of soil. Might have actually been more, to be honest with you. And at one point, this particular job had 27 different soil profiles. That's how complex it was. It was right on a watershed. It had a riparian system. It had a swamp system. It was right on the watershed for the entire water source for Connecticut.
Wow.
So talk about permitting and just I had more inspectors coming in and out of there on a daily basis. It was insane. So, I had to build literally all these different soil systems to support the plants. And then, of course, the architects want to plant a swamp maple and then surround it with boxwoods, which don't want their feet dry or feet wet. And the maple wants it wet. So I had to do like clay layers to prevent the moisture from the maple tree getting into the roots of the boxwood. And,
wow.
I mean, it was just nuts. But during that whole process, every time I put more than 8 inches of soil down it, I would always put another layer of inoculant, and shit just took off like nobody's business.
Well, this is why they call you a soil engineer, Leighton. That's some intense soil building there.
Well, it all started because I got tossed into these engineered soil systems that they couldn't support the grass. And the only way that I could get that soil, which was 99% sand, 1% organic matter, to hold protozoa and fungi, was to apply a highly concentrated biological humic acid that I was been making down at Rodale. So we'd apply that to these sand systems, and the grass would and seed, and the grass would set hold and come back. And within a couple of weeks, we would have 4 to 6 inches deep of dark, rich, not completely aggregated, but on its way soil out of sand. So yeah, there's ways of doing this stuff, but again, it's all a matter of like understanding the parameters of what soil you're using and how best to use it. And I'm going to get into depth about like these super soils and or soilless mediums, and how there's hardly any nutrition. So how are you going to set up a advanced soil food web in something that's just a substrate with very little surface area for things to attach and build biofilm? The real action is going to happen down in the A layer until that soil system matures.
All right, we've got about 8 minutes left, so time for just a few more questions. Somebody was asking, why can't you collect AMOs by the ocean?
You could in where the river meets the ocean, for sure, in a brackish area. I just wouldn't collect. I wouldn't collect true ocean AMO, because I'm not sure that you would actually be able to transfer those, because they are in a more saline. I mean, the parts per million not much, but that's kind of like jumping a little bit too far over or out of the box. When we deal with salmon farms, we have to desalinate before we can even start to deal with the manure, and that's a big problem. That's a whole nother process. So no, I would not collect from an ocean, but I would definitely not hesitate to collect from a brackish water system. I believe those would be able to transfer into a soil, a terrestrial system.
All right, great. So somebody's basically asking, can you propagate AMO? Can you multiply them, or are you just stabilizing?
Yeah, absolutely. Absolutely. You stabilize it first, because you don't want to multiply the bad guys, right? So that was that little trick. Again, I'm a big fan of cone bottoms. You can buy them on US Plastics. I think they have a 15-gallon one. It's really inexpensive. You can also go to Lowe's, and Lowe's sells those 5-gallon water jugs that you flip upside down on top of your dispenser. That's a cone bottom. So all you have to do is get there's a plumbing piece that's basically has 2 ring clamps. And so you take one of them and put it over the end of the bottle, the neck, and the other one you put in a fitting, what's it called? The barbed fitting for your hose. And then all of a sudden now you take the hose, make sure it goes up over the height of the water level, and then plug that into your aerator. If you leave your air pump below the top of that water and that air pump goes off or you lose power, you're going to blow your air pump. So you always got to make sure that air pump is higher than the top of that water. But anyway, now you've directly connected air to a water bottle. You cut the top of it off. The bottom of it normally will now become the top of it. And there's a homemade, inexpensive cone bottom brewer. So again, you add your AMO to the water that's in your brewer, a tiny bit of molasses until you understand what you're doing. And you really should have a microscope at this point because if you add too much molasses, you're going to screw up the cycle. And I usually deal with 125-gallon brewers. I don't do a lot of 15 anymore. The last time I did a 15, I think I used a quarter teaspoon of molasses for 15 gallons. So at 5 gallons, that's just a couple of drops. And again, 30 hours. Do not go over. Do not go under. 30 hours, you got to be die hard and get it back out. Get it out onto the soil or into your pots.
All right. Cool. I think that answers. Somebody was asking to talk a little bit more about stabilizing the fish waste, but I think we've covered that pretty well.
Basically, like Chris said, the nose knows. All right. So unless you have a microscope, it should smell sweet. All right. Sometimes, like the last order I got from Keith, shipped before Christmas, and it got hung up in the Christmas traffic. So by the time I got it here, it was 12 days old, and the bucket had slightly swelled, which I knew was going to happen. So basically, that is the off-gassing of hydrogen sulfide. So all I had to do is just carefully crack the bucket so the shit didn't blow everywhere, and then put it into the brewer and start stabilizing it. And I think it was definitely by the next day. It might have even been by the end of the day that I could start to smell that sweet smell. But definitely by the next day, all I could smell was the sweet smell. So I knew I was in the right spot. Put it under the microscope. Yep, everything was good. So I know that by being careful, brew it till it starts to smell sweet, and then brew it another day.
Okay.
And if it doesn't turn sweet, that means you're not pushing enough air through that. You got too small of an air pump. I would err on the heavy side with the air pumps. A little bit more is better. When I first went down to Rodale, that was like the second thing that Elaine was like, well, now that I know you're so smart, can you come over here and fix my brewers? And I'm like, all right, what the fuck is a brewer, right? And so she showed it to me, and I'm like, oh, well, that's like what I use to stabilize my fish shit. And I'm like, what's the problem? She goes, well, I just can't get it to make good tea. So I looked at the pump size and I looked at the cone size. So, well, your cone is too big. There's a formula for this. Width and height are critical versus CFM, how much air is being driven. So if it's too tall and not wide enough, you're not going to get agitation and you're not going to get airflow, CFM. So I always tell people, buy a bigger air pump than they think. I mean, don't buy one that's so big that it's going to just blow your water all over the place. But definitely on a bigger side is better, because that's going to push more DO through it. And if you do end up with a taller water column than wider, you're going to still be able to function, and you're not going to have issues. Because you don't want to buy two. It's better to buy one that's a little bit bigger.
So so we've got just 2 minutes left, or really 1 minute. But somebody just asked, what's the largest size brewer? So you were kind of answering that, but what is there a max size that you would go to?
I don't really like going over 125, because I can make a hardcore inoculant at 125 and then add 10 gallons of that to another 100 gallons of water and repeat. So how many? I need 1,000 gallons in a 125 gallon brewer. The bigger the brewer, the more complicated it is, the harder to clean it is, the bigger the air pump you need. It just gets to a cost of scale that's not really fathomable. I mean, I know people that have 250 gallon brewers, and it's just a nightmare. It's just too complex. So go 125. There's plenty of air pumps that are available that are big enough to handle that. Again, US Plastics has wonderful 125s. Then you get a metal stand with them. So that would be what I'd recommend.
Awesome. All right. Well, thank you, Leighton. It's been a pleasure, as always, for me, and I'm sure for everybody else here. Just want to remind everybody, Leighton is the genius behind this event here. He put all this together. He brought all the speakers together. So we're really appreciative of him for bringing this education to everybody.
Dude, I'm hardly a genius. I'm just a lunatic on the bridge. I'm bringing education to the world. Do everything I can to give back before I'm gone.
Yeah, well, everybody here appreciates it a lot. So thanks everybody for joining us today. So we'll start again tomorrow at 9 a.m. First session, 9 a.m. That's 9 a.m. Pacific. check your emails for the reminders. You should get one 15 minutes before the session and be getting one today as well. So we'll see you all tomorrow. Thanks a lot.
Have a good night.